Okay, I think it's time to start again. Let me see, where are we now? Oh. Okay, so now I'm going to talk about some efficient strategies to find the area of interest. So that's about tiling and for autofocus, this little lecture. If I manage to move from one slide to another, it will be even better. Here we go. So sometimes we have to choose an objective that has a smaller field of view. And it's very useful to be able to use tiling or mosaic or an overview or something like this. And the way it works is that we take one image and then we move and we take another one next to it and another one, etc. And we keep on going like this. And then the software assembles all the images together. And that is very useful to be able to image over a much larger field of view than what the objectives allows us. So this is called tiling or mosaic or overview, something like this. It has many functions. One of them is that you can actually look, so you can explore your sample and navigate in your sample using the XY automation of the microscope. And so, for example, in here, we're going to acquire a whole image, an image of the whole sample. And then we're going to, so we have a very good overview of how the sample is. And then we can log positions inside the sample and then revisit them afterwards. And so for example, here we're going to use a low magnification, maybe a 10x objective. It's an air objective. We don't need to put oil or anything. And then we use a minimum number of channel. Maybe in here we could even have put red only, but we need to see the edge of the tissue. So there are some cells a little bit everywhere. And then we use no averaging, full field of view, and then we get this very big image, which is our overview. It's a low resolution image, but it shows us what we want, which is, where's the red? And then we can save this image as an overview to then show, I've imaged this high resolution here, I've imaged this high resolution there. Over a little bit, slide. And then, so for example, on a single point confocal, we would use large pixels. So we know we are undersampling. We will see this next week, but it doesn't matter because all we want to see is the overview of the image. We use a large pinhole because that allows us not to bleach the sample when we make the overview. We can, if we have a large pinhole, the image is brighter, so we can bring down the laser power, for example. We can use a bidirectional scan, so even if there's a little bit of shift between the lines, it doesn't matter. And we can use a resin scanner if we have, and we can use a fast speed, so a short pixel dwell time. All this we will see next week. And if we have a camera-based system, we could bin the camera. This is also something we'll see next week, to use big pixels, and we can use then a low illumination, and the images will be noisy, but never mind. All we want to see is the overview. Okay, so once we get the overview, we can often, I mean, in any software, any microscope where you have an automated stage, you can do what I'm talking about now. Okay, so you can take this big overview and then afterwards you can click in the image and save different positions. So you can say here, I save position number 1, and then I save position number 2. And you make a list of positions of areas in your sample that are interesting. And then afterwards, you go back to each of these positions after changing the parameters on the microscope. Maybe you're going to use a different objective. You're going to use like a higher sampling, so smaller pixel size. Maybe you're going to do a Z stack. You put more colors, etc. So we're going to revisit these positions that we have found in the big overview and that were potentially interesting, and then we go back there and we image. And we're able, then when we publish, we're able to say the position 1 and position 2 are in these areas of this big overview. And then we can also log all the positions that are interesting. So this way to navigate, instead of looking in the eyepiece at your sample and just bleach, 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 we first take an overview and we use the overview to navigate in the sample. Like for example, in here, once I have this overview, I can actually click in the software and say, move the stage to this position. And that can be done again on any macroscope where you have an automated stage. You can do that, right? So you can use this overview to navigate and move the stage to these places. Okay, all these type of things I will show you when we start the workshop tomorrow, Fridays and Mondays. I will show you how to do this, but remember that anyone who has an automated stage can do what I'm showing right now. This is a very powerful way to navigate in your sample. So any system that is automated in XY can do this sort of things. We can even analyze things in the meantime. So what we can do is that we can take this very low. This is an example of what we did with yeast, where we take very low resolution images and automatically analyze the areas of interest. And we find automatically when we had like this mother cell with like pink and then something green next to it, this was an interesting pair, and then the others were not interesting. So we analyze the image to find this, and the positions of the interesting objects are automatically logged into the software. So we don't even have to select positions. They come up, the software will come up with analyzing the overview image and finding the interesting positions. And then we get a log of positions, and the settings are automatically changed and everything is re-imaged in such a way that we then get a high resolution image like this with all the colors that we want and everything. And you see how this image has been analyzed automatically. And so while it's doing it and imaging just the stuff that you're interested in, so you only get the images of the things that you're interested in, this is very useful, then you can go for a coffee, which is not bad. So use the automation of the macroscope. That's the reason why we have such advanced macroscopes, is because you can go for a coffee. You don't have to sit for hours and hours there. So I will show you a little bit how to do this when you come to me. One other thing that is, when it comes to automation, that is very useful is the autofocus. So the focus is very important. When you have a long sample like this one, I think, I can't remember who, but someone is imaging a skin, so you will recognize a long strip of skin in here. And what you can see is that the sample is in focus in here, and you take this big overview image, and then the focus fades as we go away, as we go to the sides. So we have a problem with the focus from one side to another, and it's much harder, it's pretty much impossible to analyze this image because of this gradient of focus. And you can see that sometimes you even get out of focus within one image. So this is over many images because that's an overview, but sometimes even within one field of view, you can get this gradient of focus. So you need to learn how to recognize something like this, that why is it, you need to learn how to see it. We have a tendency to just brush away the things that don't work. But when you see this, there is clearly a problem with the sample. Maybe the sample is not straight on the stage or something like this. So you need to learn how to recognize this type of things. This makes it impossible to analyze, and also the intensities are wrong. You can see there is a gradient of intensity. So the focus is very important. We need to have a focus strategy. How can we make sure that the way we find the focus that we're going to set as the focus of our image is that reproducible between samples? And also, how do we maintain the focus during the imaging while the microscope is imaging? So these two questions are very important. So there are different reasons why we need an autofocus. Sometimes time-lapse, yes. If we image over a long time, we definitely need to make sure we keep the focus. And people think about autofocus only for time lapses, but actually it's really wrong. We use the autofocus all the time, even on fixed samples. For example, if you take a slide that was in the fridge, so your sample is cold, and you want to image with an oil objective, you're going to get a very fast focus drift. Within like seconds, basically, you see it, you image, you come back. 10 seconds later, you image again, and your sample is slightly out of focus. And that is because of the oil, the transfer of energy within the oil. And so a cold sample with an oil objective, you get a focus drift for sure. Another reason why we need to use an autofocus is when we image over long distances. For example, if we have a big sample, like a section of a brain or something like this, we have to image an overview, and there it can be out of focus from one side to another. Or if we have a multi-well plate. And sometimes we need autofocus because the surface of our sample is not flat. For example, if we have a gel, the surface is never flat, and then some cells on top, then we will have this problem with the focus. So we need autofocus in that case. So the major cause of loss of focus is the temperature drift. And this is what we have where our sample is cold and we image with an oil objective. So temperature drift, it can be that the sample is not flat. And this can be due to a bad sample mounting, so the cover slip is not flat. It can be also that the sample does not sit snugly on the stage and that it's not flat, or you will see that it can be a problem with the sample preparation. We see this on Tuesday. And it is not rare that the bottom of multi-well plates is not flat, unfortunately, so it is not uncommon. When it comes to keeping the sample in focus during the imaging, this is critical for image analysis. And that means that you will have, if you have a correct and stable focus, you have reliable intensity information, and you have also reliable shape information. So it's really important to have a correct focus. The solutions that we have to get the focus correctly and to keep it are hardware and software autofocus. Also, we can use a smaller field of view if we have a problem like what I've shown before. If within one field of view we have one part that is in focus, one that is not, then we have to reduce the field of view. And we can also clearly add a small Z stack to catch all the different Z levels if it's within one field of view. So I'm going to present here the hardware and software autofocus so that you know the difference. So the software autofocus is basically, it's also called, I don't know, I think I've heard laser image autofocus, that's it. So basically what happens is that we're going to take lots of images of the sample, and the one that is most in focus, so the software will analyze these images and decide this one is the most contrasted. So the one that is most contrasted is going to be chosen as the one that is most in focus. So it is best to do this with low numerical aperture objectives. Because of what I said, with the high numerical aperture objective, they have a small depth of field, and that may confuse the software. So it's best to do this with low numerical aperture objectives. The disadvantage of using a software autofocus is that we are imaging our sample, and therefore we're bleaching it. Also, it takes a little bit of time. So if we need to do this every time, for example, we take a tile from a mosaic, is going to take significant time in the imaging time. And also, it doesn't work if we have a very dim signal, because we need to have a certain contrast to be able to, for the software to be able to identify which image has the best contrast. And it doesn't work if our sample is slightly three-dimensional, and therefore we're going to have in this image some things that are well contrasted and in focus, and in this image and in this image, and all these images we will have some objects that are well contrasted, but not exactly the same objects in all the different Z levels. So if we have an object that is slightly 3D and one can say that, yeah, many objects are very 3D, then we have a problem. So, for example, if you have vesicles inside cells, the advantage of the software autofocus is that it's available on any system, and that can be inverted or upright systems. Now, if we look at the hardware autofocus, it works in a completely different way. So I think that one is called also laser autofocus by some companies. So the way it works is that here we have our cover slip, and then we have cells on top of the cover slip, and we say, hey, this is the focus that I am interested in. So we say we would like to be more or less at the equator of the nucleus. We said that's the focal point of interest. So there is a distance between the top of the cover slip and the plane of interest. This is called the offset. So what happens is that the hardware autofocus works with a little LED that is on the side, let's say, of in the microscope, and it sends some very long wave.
What, excuse me?
Sorry.
Yes, it's right. Okay.
Sorry.
Mm-hmm.
Christos?
Sorry.
Okay. I think that Christos was talking, but now he's muted. Christos, if you want to talk, you're welcome.
No, I was not talking. I'm sorry.
Sylvie,
Right.
hello.
Yes.
I have a question. How can we know if our microscope has a software autofocus or a hardware autofocus?
Good question. So I think you need to ask because so software autofocus, if you have a microscope that can do a Z-stack, it is quite likely that you have software autofocus. Sometimes this comes as an extra, like an option in the software. That's possible. And then you have to figure out if you have bought the option or not. So that's something I cannot tell you, but you need to ask the person in charge of the microscope if you have it. And if you nobody knows, then you need to call the company and ask them, do you know if the system has? And they will tell you at least where to find. For the hardware autofocus, it's a little bit the same thing. So the hardware autofocus always comes as an option on the microscope. So this is not standard. You will, it depends on from one microscope to the next, even if it's exactly the same model, you may or you may not have it. So you need to ask the person in charge and ask the company if nobody knows.
Yeah, we have it as an option in the software.
If you have this option, then you can use it. So one of the assignments that you have.
We are using the same microscope system, so I know that we don't have that for hard drive. We don't have autofocus on hard drive.
You do not have.
No, we have that on software, but we don't have on the hardware, so we cannot use that.
So how do you know that you do not have it?
I asked the person in charge.
Yeah,
so
actually,
this is the way, pretty much the only way to know, really.
Yeah, and actually we don't use it because it doesn't work well, so we don't use it.
So they are. You mean the software autofocus?
I guess it's software autofocus. Yeah.
So this is something that is it works very well for some samples and not so much for other samples. And also you need to learn how to use it. So it may work quite okay. It may, for example, allow you to find the sample, but not exactly the plane that you want. That's the problem. If you have, for a software autofocus, if you have several Z levels where you have something that is in focus, then the software autofocus is not going to work. So because it's based on the contrast in the image. So if you have several images that are in contrast, then it's a problem.
Yeah, that is what we.
Yeah. So this is one of the assignments that you all have. You will have to figure out if you have a hardware and software autofocus and also how it works if you have one.
I have
Melissa, you had a question?
Yeah, I have a question. I would just want to confirm, so if in case I would want to image vesicles in a live sample over time, like 6 hours or something like that, then I can't use autofocus, or is it possible?
Yes, you can.
Oh yeah, I can.
So yes.
Because I asked this from the person responsible, and I don't know if he just didn't want to work because he was like, no, you can't. You
But
have
it's
to.
possible. It depends what you have. So if you have a software autofocus,
Okay.
if you have a software autofocus, or do you know if you have a hardware autofocus?
I don't know which one.
I don't know. So you need to ask. So
Yeah.
basically, typically this type of questions where you're going to be imaging vesicles, you're going to want to use a high numerical aperture objective because then you need a high resolution. The vesicles are often crowded, so you need to differentiate them. So you're going to use a high numerical aperture objective. In live samples, please use a water objective. And then that means that you have a thin depth of field, and that is going to trick this is the typical thing where the software autofocus fails because you're going to have vesicles in focus at different Z level when you take the Z stack. So the only thing you can do to compensate this tech when you do the autofocus, the only thing you can do to compensate is really to take a Z stack.
Yeah.
Which is not very good for live sample imaging. If you're imaging with a wide field system, I don't suppose you do that. I suppose you use a point confocal, right?
Yeah.
So that's even worse. One thing that can be done is because it's not very good with live sample anyway. If you have to do a Z-stack on top of your normal stuff, for this type of applications, definitely the hardware autofocus is best.
Okay.
It's really very reliable because the thing with the hardware autofocus is this light that comes in is a very long wavelength, is around 860 nanometers. So it does not, number one, it does not cause any bleaching of our fluorophores because it's a very, very long wavelength. And also it's not very it doesn't give a lot of photo damage because it's a long wavelength, low energy, right? So, so this is really the best option, is to get a hardware autofocus. What is your system?
Stellaris 8.
Right. So I think that Leica has also a solution for that.
Okay.
You can ask for a quote and
Okay.
see. But it is a very useful tool,
Yeah.
really.
Yeah.
May I add something?
Yeah.
I think it's also important to consider that if you are using a water objective, it's very important. When the water evaporates, you are doing live imaging, so you are in a closed enclosure, so you have to be very careful that you always have enough water immersion on your objective. Otherwise, even a little bit, if you have less, it will influence the focus and how especially the software
Okay, so is the
autofocus.
max like then maybe like 1 hour, 1-2 max, like
Oh,
before
no,
you...
even 1 hour. It's very small volume, that you put on the objective.
Yeah.
So you have to have a special, you might have something that all the time fills in, so it keeps it for a long time. Because if it's not enough, and poof, it will not be, you will not be able to use it.
Then you lose the focus completely if you lose the water, and then it's really your whole experiment doesn't work. But you can either use, like Gabi says, something like a machine that refills the water. That exists. A water dispenser is called. But you can also use, in some cases, certain objectives are designed to, so certain water objective, you can put a special oil that has the refractive index of water. And that oil does not evaporate, but it anyway the same refractive index of water. So what you need to do is ask Leica if they have such an oil. And then you don't have to bother with is this evaporating or not, but you anyway have the correct objective.
Okay. As long as you don't have to go between wells or long distances, because otherwise you to smear everything. So that's also something
if you have
to
to
keep
travel
in mind.
a lot, then it doesn't really work with oil. So this is all the type of compromises that we have.
In one point, it works very well, as Sylvie said, and we use it ourselves. We used it, but if you start to have to move between wells, then with oil you might be in trouble. Then it's better to have a water dispenser.
Okay, so like so far I haven't moved. I'm in the same. Like,
you can stay in the same place, but use then some oil on the water objective. But you need to ask Leica.
Okay, thank you.
If they have such an oil, you explain to them that you don't want the water to evaporate. Good. And then I think I'm almost finished. I'm just checking the time. Yes, okay. So this here we have a very long wavelengths, and what happens is that so because we have a cover slip in here, and here we have water. For example, this sample is in water. Then we have refraction. And so we have spoken about the refraction, and what happens when there is a drift in focus. The software, thanks to this little light that is coming back down and then detected, will find exactly the position of the distance between the objective and this refraction index interface, right? So in this case, if we have water in here, the refraction index interface can be between the top of the cover slip and the sample. So we have then the refraction index interface. The software calculates the distance between the cover slip and the objective and corrects it. So you are going to set an offset, and you say, this is the top of my cover slip, this is my plane of interest, and clearly you use the correct cover slip, and then the software will calculate and move the objective as if you were focusing. So it moves the objective to always keep the same distance between your plane of interest and the top of the objective. And so this does not interfere with imaging at all. So this process can be in the background while you're imaging, you will not notice it. And then this hardware autofocus will probe the focus every like 200 milliseconds or something like this. It will check that the sample is still in focus. And if there has been a slight focus drift, it will move the objective to catch up. So this is extremely useful to be able to keep the focus without interfering with the imaging at all. And you can do that while, so it's automatic, so you don't even need to bother, you just turn it on and then it's automatically done. The disadvantages of hardware autofocus is that it requires a refraction index interface. So if you have a perfect sample where the immersion medium of the objective matches completely the refractive index of the sample, then there's no interface in here, so there's no refraction and the autofocus doesn't work. Okay, so unfortunately, it only works if the refraction indices are not matched. So it also only works if the focus of interest does not move. So if you are in a situation where you always have the same distance between the top of the cover slip and your plane of interest, then it's fine. For example, for you, Melissa, if you say, okay, I'm going to look at how the vesicles are moving around. I'm going to look at one plane, but it's always going to be the same plane. You're going to have you consider that everywhere in the cell, it's going to be always the same. For example, you say, okay, I'm going to go 2 microns above the cover slip, and that's where I'm going to look at the vesicle trafficking, and I'm just going to stay there. Then this is a perfect system for you, because that will work. But if you have something where you would like to track the vesicles, and they move up and down in the cells, that doesn't work. Because you cannot follow up and down. You're always at the same distance to the cover slip with this hardware autofocus, okay? So any wavy tissue section or cells on the surface of a gel that is not flat, the hardware autofocus will not work. On top of that, it is not available on upright systems. It's only available on inverted systems. The advantages of hardware autofocus is that it's instantaneous. It just goes on without you knowing it in the background. It doesn't bleach the sample because it doesn't excite the fluorophores. And they are many applications, I guess. When the sample is at a fixed, it works when you have applications where the sample is at a fixed distance to the cover slip. This is when it works. And there are many, I think I'm missing the MA. There are many applications that way. So in the Nikon system, the hardware autofocus is called perfect focus system. And I hate them for that because I keep confusing PFS with point spread function. PSF. So if I say PFS instead of PFS, you know why? It's because of Nikon. I can blame them. And then Zeiss, their hardware autofocus is called definite focus. Leica is called adaptive focus control, AFC. And Olympus is called ZDC, zero drift control. So if you find one of those on your microscope, then you know that you have a hardware autofocus. And you can go and ask the company if you're not sure. an extra layer,
Exactly.
if you want to say, that we can do to keep the focus, and that is slanted or complex surfaces. So how do we do if we have a gel and the surface of the gel is not flat and it sinks like this? Gels always do that. So either they are in this way or they are like a little mount. What do we do to image the surface if we only want to image the surface? So with this system of complex surfaces, you could go in the software and focus in here, and you record the XY and the Z position, and then you go to another place and you record again the coordinates and you do this several times across your whole sample, and then the software will calculate an imaging surface. And it will extrapolate basically between your point. And that means that if you go now to a place where you have not logged the position, it will find the correct focus, hopefully. So this depends on how complex the surface is. The more complex the surface, the more reference points you need to have. But it works quite well anyway. So the disadvantage of this complex surface focus is that you need to enter the position manually. And also if there's any drift, this is not compensated for, right? It stays at these positions, but it doesn't follow if they change. The advantage is it works on uneven surface, even sloped surface like this one. And it also works for perfect sample where you have no refractive index interface. And it works if the sample is not flat. So if your sample is a bit wavy, then it works as well. So it's very good. So has anyone used this type of slanted surface?
No.
No? No one? Someone has a hand up? Sophie, have you used it?
I think it's a similar problem, but we don't have a gel, but we have the large chip and we scan across using over time. And to compensate for the tilt of the chip, I think we are using this approach.
Right, yes, that would be perfect for this one because your chip would be anyway sort of flat. It's not like a gel that really goes up and down. Yours would have long distances, so with only not so many points, you can get a pretty accurate surface.
Yes, I think there's a module for it in Zen where you can choose 3 or 7 points and it calculates the tilt.
Good. Do you know what it's called?
No, no, I don't remember. It's this advanced setup part of Zen.
Yeah, so I think this is very useful because if you can send me the name, I can put it in Canvas, and people can Google and try to figure out how it works. Because if you have the module on your software, because this is now a software-based thing, so if you have the module there, you can just use it. So that would be very useful to know this, and we can share with everyone.
Yes, happy to send it over.
Thank you. But again, this is like an option. So, but if it's a software thing, it's maybe not as expensive as a hardware. So you can probably get it as well if you need it. Great. So please use the automation of the macroscope to explore your sample and find the area of interest. If you just look with your eyes, first your eyes are very good with green, but not good with the other colors. And then you bleach your sample without getting anything out of it. So instead of that, the strategy is that you go to your microscope, you focus the first thing, and then afterwards you make a big tiled image, and you use this tiled image to navigate. With the image you navigate, you move the stage by clicking in the image to go to that place. And this is again available on any macroscope that has an automated stage. So you have less bleaching, it takes less time, and also you have less risk of imaging twice the same area because now you can put numbers on them and go from one to another systematically. It's very important to set a reliable focus so that you get reliable image analysis. So you must establish a focus strategy. I think. I can't remember when it is. Well, you have to do that. So the questions that you want to ask yourself are, what will you focus on to ensure that images of different samples are comparable? Even if you have one cell layer, if you are looking at small objects, you will have a completely different image if you image sort of the first third of the inside the cell or the last third inside the cell. So it's really important to define for yourself what you want to focus on so that you apply the same strategy for all your samples. And then how will you ensure that the focus is stable during imaging? Sometimes we even take like a big image and it takes quite a lot of time to image. So we need to have a focus stable during that time. And then we need to focus on a structure that is not affected by your different treatments. So if, for example, you say, I treat my cells, I have treated and non-treated samples, and then the treated sample is very flat and the non-treated is very rounded, it's going to be difficult for you to focus with the same strategy if you do not have a proper strategy where the tooth, you find something that is not modified. For example, you can say between the sample and the treatment, for example, you could say, I'm going to find the top of the cover slip, and then I will always go 3 microns higher. And that is a strategy, okay? Or I will always find the filopodia, and I will always focus on the filopodia. That is a good strategy. Or I'm going to focus on the equator of the nucleus. But define something for yourself and stick to it so that you ensure reproducibility from sample to sample. And then you should write your focus strategy in the material methods in article so that other people can reproduce their data. And if it's possible, use a hardware autofocus because this is really an excellent tool. Right, so I think that now I'm going to stop sharing and let me see where we are. That's it, right? I'm spot on time. So what we're going to do now is that we're going to go to the group discussion and we're going to talk about the focus strategy. I know that it was coming, but I couldn't remember when. So you're going to have to discuss now your focus strategy, and then this is one of the portfolio things, and then we'll go to group some quizzes. So now we're going to leave this Zoom, and I hope that people who are online will join us again next week. But right now, let's go to the other Zoom. Bye, everybody.